So building on that primary structure and going to the other levels of folding, we're going to look at secondary, tertiary, and quaternary structures. So the shape of an alpha helix is similar to that of a spiral staircase. The alpha helix acquires a coiled shape from hydrogen bonds that occur between the oxygens of the C to O group, the carboxyl group, and the oxygen of the NH or amine group in the next turn. So we're going to learn how to be able to describe secondary, tertiary, and quaternary structures for the protein and how we can unravel or denature them. So the secondary structure of an alpha helix is what forms when hydrogen bonds form between the oxygen of the CO groups and the nitrogen, the hydrogen of the NH groups of the amine group bonds in the next turn of alpha helix. The formation of many hydrogen bonds along the polypeptide chain gives the helical shape of that staircase. Okay. So also for your secondary structure, you have beta pleated sheets. So these are where, again, hydrogen bonds are going to form between the carboxyl or the carbonyl oxygen atoms and the hydrogen atoms in the amide groups, bending the polypeptide chain into a sheet. Okay, so keywords for your secondary structure are going to be those alpha helices and your beta pleated sheets. So you can also have a triple helix, and this is where 3 polypeptide chains are woven together. And the hydrogen bonds hold the chains together, giving the polypeptide the added strength typical of collagen, which is connective tissue, skin, tendons, and cartilage. So things that are a little more tough. Okay, so we have this triple helix as well. Your tertiary or third level of structure, this is an overall 3-dimensional shape that's caused by interactions of different parts of the chain, causing it to bend and twist. This is determined by crosslinks and the attractions of repulsions between the side chains or the R groups of amino acids in the polypeptide chain. So remember, primary and secondary interactions, those are going to be backbone interactions, where your tertiary and quaternary structure, those are going to be your sidearm interactions. Okay. And proteins can become functional at the tertiary structure. For example, myoglobin only stops at the tertiary structure. It does not go on to have a quaternary structure. And so proteins can start to become active at that tertiary structure. And so again, the interactions between the amino acid side chains, or the R groups, fold a protein into the specific 3D shape, and that is called the tertiary structure. So sections of the protein can interact to create a tertiary structure of a protein due to hydrophobic interactions between carbons, right? Those 2 nonpolar amino acids. Hydrophilic interactions between the external water-loving amino acids, the polar amino acids. Salt bridges, which are ionic bonds that are ionized R groups of basic amino acids groups, as well as hydrogen bonds, which are between the H of the polar R group and the O of another, or nitrogen, right, of another amino acid. And then we have that specific special disulfide bond, the SS, that is between the SH groups of cysteine amino acids. The reason it specifically says cysteine, you don't have to memorize all the amino acids. I had to back in the day, but I'm not going to make you do it. But the disulfide bond is a special bond. And so cysteine is the only amino acid that has sulfur on the outside. So that's why I ask you to learn him. Okay, so then link this to health. What is sickle cell anemia? Well, it's caused by an abnormality in the shape of one of the subunits of the hemoglobin proteins. So when we have a normal beta chain, we have glutamine here, right? And this is polar. But then in sickle cell, that is mutated to valine, which is nonpolar. So you have something that normally loves water, and then you're sticking a nonpolar guy that's terrified of water on the outside. Well, he's going to look for other amino acids that are on the outside, other carbon amino acids, and that are hydrophobic. And this is called aggregation or like clumping, right? Because they're basically looking for other carbons or other hydrophobic amino acids to grab onto because they're so scared and they can't swim, so they're kind of like drowning, so they're like, help me, right? So that causes the protein to misfold. If it misfolds, it misfunctions. Okay, so in this, for specifically sickle cell, it's the 6th amino acid in the beta chain. The polar acidic glutamic acid is replaced with valine, again the nonpolar. And then it's attracted to the nonpolar within the beta hemoglobin chains, that's the aggregation. Then the red blood cells are going to change from the rounded shape to that crescent sickle cell shape, which interferes with your ability to transport enough oxygen. So, these hydrophobic interactions also cause sickle cell hemoglobin molecules to stick together, and they form insoluble fibers of sickle cell hemoglobin that can clog capillaries, cause inflammation, pain, and organ damage, as well as cause low levels of oxygen in the affected tissues. Because remember, your red blood cells, those are going to be transporting your oxygen using that hemoglobin. If we have misshaped blood cells, it's not going to be able to transport oxygen as well, and therefore it's going to cause oxygen delivery issues, therefore causing low oxygen levels in the affected tissues. Okay, so let's do a little study check, and let's indicate the type of protein structure of A, B, C, and D. Okay, pause me and come back. All right, so a polypeptide chain that are held together by side by the H bonds, that's going to be the beta pleated sheets. The sequence of amino acids, think of that pearl necklace strand, that would be your primary sequence. That corkscrew, that's going to be your alpha helix. And then with the 3 peptide chains, 3 would be triple. That would be your triple helix. So select the type of tertiary interactions between leucine, valine, 2 cysteines, aspartic acid and lysine, and serine and threonine. So again, look at your pictures so that you can see which ones are which. Pause me and come back. So leucine and leucine, those are 2 carbon, hydrogen, so those would be hydrophobic. Remember that cysteine, that's the special guy, and he has an SH on the end, and so 2 SHs come together is the disulfide. Aspartic and lysine, that would be ionic, where serine and threonine would be those hydrogen bonds. Okay, good job.